Welcome back. In this video, we are going to explore the impact microbial enzymes and other enzymes make on your life. I think you will be surprised at the number and frequency of the use of microbial enzymes. Let's get started. I have provided an outline here of this talk to help you better see how it all fits together. Before we get into the video in detail, I wanted to demonstrate how big the impact of microbes and their enzymes have had and still do have on human society. I will do this by asking you a question. Why did humans move from a hunter-gatherer society to an agricultural society? Early humans lived in small groups and foraged for plants and animals that they could eat. About 10,000 years ago, humans started to domesticate animals and plants and focus on growing just a few of them. Why did they do this? Write down an answer and I will give you my favorite theory at the end of this video. And of course, it's gonna involve microbes. For you to understand the impact of microbial enzymes, you need to spend a little time thinking and learning about chemical reactions and enzymes. If you are uncomfortable with chemical formulas, I suggest you view the video on chemistry and bacterial structure. For our example, we will use the first step in glycolysis, a process that occurs in your cells continually. Glycolysis is a series of reactions that the mitochondria in your body use to break down sugar and convert it into energy that your body can use. In a typical reaction, the substrates, in this example glucose and ATP, combine to form the products, glucose 6-phosphate and ADP. For this reaction, phosphate attaches to the 6th position of the glucose molecule. If concentrations, temperature, and pressure are kept constant, the reaction will reach equilibrium, meaning the concentrations of substrates and products stays about the same. You can visualize chemical reactions as molecules bumping into one another in just the right way that they react and form something new. This concept is much easier to see in an animation, so let's go ahead and do that. We have here another common reaction, that of acetyl-CoA and coenzyme A. Don't worry too much about what they are. Pay attention to the idea here. First, let's leave the concentrations at 10 and see what happens. After 30 seconds, you see only one reaction. That acetate and CoA bump together in just the right way to react. Just for fun, let's increase the concentration of both to 50. Now they don't have to be the same concentration, but I'm just going to do that for simplicity here. Now there is 5 times as many molecules to react. In 30 seconds, we saw 20 reactions. So, increasing the concentration increases the reaction rate. This idea is generalized as Le Chartier's principle, which states that given a reaction at equilibrium, a change in concentration, temperature, or pressure will cause a reaction that shifts the equilibrium in a manner to lessen that change. If we go back to our reaction with glucose, and we increase the amount of glucose, that will cause the reaction to occur more quickly, creating more glucose 6-phosphate. The more rapid rate of reaction continues until the concentration of glucose drops enough that the reaction slows because either the ATP is used up or the glucose and ATP can no longer find each other and react. Conversely, if you increase the amount of glucose 6-phosphate, this will inhibit the forward reaction because it will be harder for the glucose and ATP to find each other in all those glucose 6-phosphate molecules. So the consequences are increasing substrates increases production of product, and increasing products slows the production of product. Another important concept is activation energy. We can graph a reaction out and represent this conversion as shown in the diagram at the right, which is called a reaction coordinate diagram. Again, don't worry, I don't expect you to remember the name. It shows how the energy in a system changes during the reaction. If a reaction is favorable, in other words, it will occur without added energy, the energy state after the reaction is lower. It's like having a ball on a hill and it will roll down the hill. When two molecules collide, they have to have enough momentum and the right orientation to collide in a way that causes a reaction. The energy necessary to produce this ideal collision is called the activation energy, and the graph shows this as a steep curve in the middle. If a reaction is favorable, you have to put in that amount of energy to get it to go over the hump and actually run the reaction. In living cells, enzymes carry out reactions. Enzymes do not make reactions more favorable. What they do is they lower the activation energy, and you can see this represented as that lower curve. It makes it much easier for the reactants to get together and do their thing. So how do enzymes do this? Let's imagine another molecular picture. At the right, you see the structure of glucokinase, an enzyme that catalyzes the conversion of glucose to glucose 6-phosphate. By the way, this structure was generated by some brilliant protein chemists who use X-ray crystallographic technology. These are brilliant people. As you can see, the enzyme is kind of globular, and it has this little pocket where glucose and ATP get together. Glucokinase captures glucose and ATP and holds them together in just the right orientation so that they'll react in that little pocket. And that's how enzymes speed reactions. Let's return to our reaction simulation, but this time add an enzyme. You will note this example enzyme also has a pocket where substrates bind and react. This pocket is called the active site of the enzyme. We will leave our numbers at 10 just like before. As you can see, after 30 seconds, we had 6 reactions. That's 6 times faster than what we saw without an enzyme. Of course, this is just a simulation, and enzymes speed up reactions much more than that. Now let's try increasing it to 50 and see what happens with an enzyme. After 30 seconds, 42 of the substrates were converted into products out of 50. Again, it makes a big difference. You now have a good grasp of what enzymes are doing in cells and why they are so powerful. Without the catalytic prowess of enzymes, life would not be possible. Let's move on to examples of how enzymes and reactions in the cell produce things of human interest. The examples we are going to cover extensively are food fermentations. Microorganisms often take nutrients we give them, such as milk, grape juice, or malt, and convert them into products we value. These conversions require multi-step chemical reactions, each one catalyzed by an enzyme. Yeast species, mostly part of this genus Streptomyces, are used in making bread and in fermenting alcohol, while lactic acid bacteria, a large group of related organisms, find uses in the production of yogurt, cheese, kombucha, and even chocolate. I bet you didn't know chocolate was a fermented product. We are now going to dive into the details. If we are going to understand enzymes and the conversions they make, it is useful to understand a little metabolism. Wait, stop, don't run away, okay? It's really not that hard. And I promise not to require you to memorize any structures. Yeah, I know structures are shown here, but you don't need to memorize them. Still here? Great. What you see before you are the interconversions that many organisms use. The vast majority of eukaryotes and many bacteria use this pathway. They oxidize glucose to pyruvate and in the process generate energy and useful starting points for making many macromolecules needed in the cell. In the first part of the pathway, glucose has two phosphates from ATP added to it, and it isomerizes to fructose 1,6-bisphosphate. Adding phosphates destabilizes the sugar molecule and makes it easier to split into two 3-carbon molecules. In the next step of the pathway, glyceraldehyde 3-phosphate has another phosphate added on. This is where the magic happens, because the pathway uses an inorganic phosphate, requiring no energy expenditure by the cell. It then recovers that energy in the next couple of steps. In the final steps of the pathway, the phosphates on 1,3-bisphosphoglycerate transfer to ADP to make ATP. So in the conversion of glucose to pyruvate, 2 ATP are spent and 4 ATP are produced. Wait, what? From what I just said, the pathway consumes 2 ATP and makes 2 ATP. Why is that doing anything? Remember that you have two 3-carbon compounds, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase, I will call TPI. As an aside, here is an excellent example of changing an equilibrium. TPI will run the reaction both ways. In other words, it's a reversible enzyme. If you put TPI and dihydroxyacetone phosphate in a test tube, TPI converts about half the DHAP to glyceraldehyde 3-phosphate. However, since in metabolism the glyceraldehyde 3-phosphate is used in the rest of the pathway, the equilibrium is pulled toward glyceraldehyde 3-phosphate and all the dihydroxyacetone phosphate gets used. The result of the pathway is energy in the form of ATP and a bunch of pyruvate. We also have a problem. We need to regenerate the NAD to continue to run the reaction. How do we oxidize the NADH that's made? Excess NADH is the big problem for organisms that do fermentations. The solution here is to dispose of the electrons on NADH by dumping them onto something downstream of pyruvate. Fermentation is the oxidation of organic compounds in such a way that the last holder of electrons is another organic compound. There are a huge number of fermentations that convert all sorts of organic compounds into simpler ones. These take place anywhere that lacks oxygen and there are organic compounds available. In ethanol fermentation, acetaldehyde is the terminal electron acceptor, being converted into ethanol. So, this is ethanol fermentation. Why do humans care about this? Of course, you know the answer: beer and winemaking. Let's talk a little bit about winemaking and beer making. Winemaking is the alcoholic fermentation of grape juice by various species of yeast. The process begins by planting and growing good wine quality grapes in areas of the world where the climate allows ideal maturation of the fruit. Winemaking grapes are much sweeter than grapes purchased at the grocery store, and getting the sugar content just right before harvest can be critical to the winemaking process. The head winemaker of the winery judges the ripeness of the fruit, and the timing of the harvest is one of the vital factors that determines the final quantity of the wine that's made. This is where winemakers really earn their money. Harvested grapes are crushed using various types of presses, and the high sugar juice is now placed into a large fermentation vessel. Often the fermentation begins with the addition of a starter culture of yeast to the grape juice. However, the winemaker may decide to do a wild fermentation, depending upon the yeast naturally present in the grapes to do the work. White grapes or red grapes with the skins removed are used to make white wine. In contrast, the fermentation vessel contains the grape skins during the production of red wines. During the fermentation of red wine, tannins and other compounds diffuse into the wine, giving it its color and a richer flavor. Fermentation in winemaking usually lasts about 2 to 3 weeks. After the initial fermentation, aging of the wine takes place in barrels, and the quality of the barrel can have a profound effect on the quality of the wine. White wine is usually aged for 5 months, while red wine will remain in barrels for 2 years. After aging, the wine is clarified, filtered, and bottled. White wines can be sold immediately, while red wines typically go through another period of aging that lasts at least 6 months, but can continue for years. Beer making is even more complex than making wine. There are hundreds of different beer styles, and each recipe is a little different. We will cover the general process here. Beer making is a 6-step process that begins with malting. The first step involves the soaking of barley in water for several days. At this time, the grain germinates and produces amylases, enzymes that degrade starch to sugars, and proteases. These enzymes are essential to the brewing process because amylase provides sugar for fermentation, and the proteases solubilize compounds necessary for the quality of the beer. After germination, the grain is then dried and crushed to form malt. The second step, mashing, solubilizes and extracts the starch and other flavors in the grain. It begins by suspending the prepared malt in water. At this time, the brewer may include other grains and carbohydrates that provide additional starch for fermentation. Incubation of the mash at 65 degrees allows the amylase to break down the starch. And then incubation at 75 degrees inactivates the amylase. After settling, the insoluble material serves as a filter, producing a clear liquid called wort. The end result of mashing is a conversion of most of the starch into fermentable sugars, such as maltose, sucrose, fructose, and glucose. In the third step, hops and wort are combined and boiled for several hours. Boiling with hops serves several purposes. It concentrates the liquid, kills any remaining microbes, and further inactivates enzymes in the mash. It also solubilizes essential compounds that either add flavor or provide antiseptic qualities that help preserve the beer. The liquid is finally ready for fermentation. In step 4, fermentation begins by adding the brewer's yeast, Saccharomyces cerevisiae, various carbensis, to the wort. The temperature and length of fermentation depend on the type of beer being made, but ranges from 3.3 to 20 degrees centigrade for 8 to 14 days. At this time, the sugars in wort are converted to ethanol and carbon dioxide, and this is where the fermentation takes place. Other compounds in the wort are also fermented, and they add to the characteristic flavor of the beer. There can be a lot of variation at this point in how this is exactly done. In step 5, the fermented wort green beer is aged at 0 to 20 degrees centigrade for weeks or months, again depending on the brewer and the beer recipe. At the time the yeast settles to the bottom of the vessel, bitter flavors mellow and other compounds form that enhance the flavor of the beer. Finishing and packaging of the beer is the final step, which can involve filtering, pasteurization, carbonation to about 0.5% carbon dioxide, and clarification. Those are the two examples that we're going to talk about for ethanol fermentation. We now turn our attention to fermentations that produce lactic acid. This is another widely used fermentation for the production of all sorts of foods. Yogurt, kimchi, kombucha, and cheese, just to name a few. One of the primary end products of these fermentations is lactic acid, hence the name of the group. Bacteria that carry out this type of metabolism are called lactic acid bacteria, and there are hundreds of different species. Lactic acid bacteria use one of two pathways. One is a homofermentative pathway that only produces lactic acid, shown here on the right. In this pathway, glucose converts to pyruvate using glycolysis, something that you've seen before, and this is in turn reduced by NADH to form lactic acid. The second pathway, heterofermentation, is more complex and uses a different glycolytic pathway, with lactic acid, ethanol, and carbon dioxide being the final end products. Heterofermentation is half as efficient as homofermentation, producing only 1 ATP per glucose fermented. And in fact, all fermentations are very inefficient, and they make a lot of product for the amount of ATP that's generated. How are lactic acid fermentations used? I'm going to just give you one example, and that is cheese making. This is, of course, done by the University of Wisconsin, after all. Cheese is a favorite food worldwide, with about 21 million tons of cheese consumed annually. There are a wide variety of cheeses, but they all rely on the action of lactic acid bacteria that ferment the lactose in milk to lactic acid. The process begins with the pasteurization, which is heating, of raw milk to kill pathogen, and also to condition the milk. An added starter culture then ferments the milk, producing lactic acid and lowering the pH. The addition of rennet, which is mostly proteases, causes the milk to curdle, forming curds. For fresh cheeses, such as cottage cheese, mozzarella, or feta, this is the end of the process. They are packaged and sold to consumers. For hard cheeses, squeezing of the curd and pressing it into a mold removes the water. This excess water is whey and is a source of whey protein. Aging in the cheese for weeks to years gives it distinct properties, and the method of aging, how much salt, the addition of starter cultures, the temperature of the room, etc., dictates the kind of cheese that's formed. After aging, the cheese is removed from the molds, packaged, and sold to consumers. Another example of using microbial enzymes is in the production of renewable energy in the form of biodiesel. Currently, our transportation system is dependent upon liquid fuel. Burning fossil fuels is increasing the amount of carbon dioxide in the atmosphere, leading to adverse environmental effects. The race is on to develop methods that create fuels that do not increase greenhouse gases. One solution under study is the use of photosynthetic algae. Algae require very little for growth, just some minerals, sunlight, carbon dioxide, and water. They then use enzymes to create energy via photosynthesis and to convert the carbon dioxide into cell structures. Scientists found that after you allow the algae to grow to sufficient numbers, if you then starve them by removing the minerals, it causes them to alter their metabolism and produce more oils. The algae are then run through a press to harvest the oil, and after extraction, biodiesel is generated that can be used directly or further refined. Creation of fuels by this process is more attractive than ethanol production using corn, since it can occur in seawater, thus freeing farmland for growing food, not fuel. It is also more efficient with the conversion of far more of the algae into usable fuel. I finished this video by briefly listing a few more areas where enzymes from microbes play a huge role in our society. Enzymes are often used to process food products. Starch is digested by amylases to produce syrups for the soft drink industry. Also, the action of celluloses can clarify fruit drinks. Detergents. Most modern detergents contain enzymes. Proteases for removing protein stains and amylases for removing starch. Bacteria are the source for many of these enzymes, and they speed up the cleaning process and reduce the temperature required to get clothes clean, saving energy. Textile and paper. Enzymes such as amylase and lipase are used in the production of paper or textiles to process materials. Since enzymes operate at lower temperatures and in less harsh conditions, their use saves energy and decreases the use of harsher chemical processes. Medicine. Some enzymes have powerful activities that find utility in the medical field. For example, the enzyme streptokinase, isolated from Streptococcus species, is used to break up blood clots, and this can be a life-saving treatment for someone suffering a heart attack. So, we come to the end of the video, and it's time to answer the question I posed to you at the beginning. Why did humans move from a hunter-gatherer society to an agricultural society? Basically, to make beer. As put forth by Brian Hayden, an archaeologist at Simon Fraser University in Canada, growing crops such as wheat, barley, and rice is hard work, yet only provides a relatively small amount of nutrition. Why do it? Cultivation began as a way of generating a more reliable food source, and part of that cultivation was celebrations and feasts at the end of the season. If you're going to party, you need liquid refreshment, and beer was an essential part of that. These gatherings were crucial to building community, In traditional feasts, there are almost always 3 ingredients: meat, some cereal grain in the form of bread or porridge, and finally alcohol. Cereal grains are challenging to produce, requiring gathering, winnowing, husking, and often grinding. You only did this for special occasions, and part of this was also making an alcoholic beverage. Feasts were more than just a meal. They were a way to create linkages between communities. If I invite you to my feast and provide fancy grains and alcohol, you will likely have a more favorable attitude toward me and will most likely reciprocate in your feast later. Beer is sacred stuff. In traditional societies, it helped to build social connection.